So now we're going to continue on with molecular biology of the gene. We have talked about replication of the DNA, and so now we're going to talk about gene expression and how that relates specifically to genes on DNA. Okay, so last week we talked about DNA replication, and the purpose of DNA replication was to allow for cells to divide, and that can either be through mitosis or through meiosis. And so we're taking DNA and we are putting it into the next generation of cells. So today we're going to start talking about gene expression. And so the purpose of gene expression is to make proteins. So we've talked about DNA is made up of genes, and so gene expression is taking DNA, making RNA from the DNA, and then the RNA being read to make a series of amino acids, which of course are the monomers of our proteins. Okay, so let's talk about some proteins you've probably heard of. So do any of these look familiar to you? We've talked about several of them. CDK and cyclin, remember, were proteins that regulated the cell cycle. Actin and tubulin are part of the cytoskeleton. ATP synthase, we know we used in cellular respiration to make ATP. Insulin helps to regulate your blood sugar levels. Hemoglobin is a protein in your red blood cells that allows it to carry oxygen to various parts of your body. So all of these are proteins, and as you know, they're super important for any kind of metabolism within your body. So where do these proteins come from? Well, they come from genes on your DNA. So the definition of a gene, it's a specific sequence within the DNA that codes for a specific protein. I don't know why I just said proton. So, for example, when we want to produce an insulin protein, there is a gene called the insulin gene, which is a specific sequence of DNA that codes for that particular protein. So we know that DNA is made up of monomers called nucleotides. And so when we align those nucleotides in specific orders, they are like an alphabet that reads for, this is the protein I want to produce. And so it's like we need a decoder. So the mRNA comes in, and it is sort of like a complementary sequencing event of the DNA genes. And that mRNA is then read by other RNAs that we'll talk about to make the proteins. And the way they do that is the sequence of nucleotides gets converted to a sequence of amino acids. So the mRNA reads the sequence of nucleotides in the DNA and then produces the sequence of amino acids that then eventually make up the protein. Okay, so let's take a closer look at RNA for a minute. So some of this is going to be a review for you, but I do want to make sure that we know how to distinguish it from DNA. So RNA is a ribonucleic acid. Remember, the RNA comes because it's a ribose sugar, and that is in comparison to DNA, which is a deoxyribose sugar. So this one has its oxygen. This is a ribose sugar. So within RNA, remember when we're looking at our bases that you do not have thymine in RNA. Instead, you have uracil. RNA is single-stranded as opposed to DNA that's double-stranded. And the nice thing is it can be temporarily paired up with DNA using that base pairing rules. And remember, those base pairs are connected by hydrogen bonds. So one thing, if you'll remember from way back when we were talking about our organic chemistry, is that those hydrogen bonds are strong enough to hold them together temporarily, but they're fairly weak bonds. It's not a covalent bond. So they can come together temporarily in a weak bond in order to move forward with our protein production. So here is our structure of our RNA. It's going to be similar to if we took the ladder apart on the DNA. So if you look along the sides of the ladder are still phosphodiester bonds, right? So those covalent bonds that link the sugars together through the phosphate group. And then off to the side, we're going to have all these bases. And that's what's going to pair up with the DNA. And just note again that we have guanine, uracil, adenine, and cytosine. So no thymine whatsoever. So remember each one of these sections right here. So we have our pentose sugar, our phosphate group, and our base pair. Our base, can't talk. Let me try that again. Phosphate group, ribose, and then our base. That is one nucleotide. So our structure of DNA is multiple nucleotides linked together through phosphodiester bonds. So here's where it gets interesting. So DNA, we pretty much have one type of DNA. But for RNA, we have 3 different types. And that really has to do with its function, not with its structure. So we have mRNA, which I've talked about a little bit. That's our messenger RNA. That's the middleman between DNA and the production of proteins. Then we have what's called tRNA, or transfer RNA, and it has what we call anticodons, which we'll talk about, that carries amino acids throughout the cell. And then we have our rRNA, ribosomal RNA, and that's found in the ribosomes and helps position the mRNA within the ribosome. If you'll remember, the ribosome is where proteins are made. So it helps position that mRNA within the ribosome so the protein can be made. And just the other thing to remember is that the ribosomal RNA is basically the universal RNA. Remember, every cell has ribosomes, and every cell has ribosomal RNA, regardless of what type of cell it is. So ribosomal RNA has often been used when you sequence it to see how closely related organisms are. So it seems like ribosomal RNA seems to be the most basic of our nucleic acids. Okay, so we've talked about the different types of RNA. Now let's go back and look at amino acids again. Let's review that. And I promise that I'm not just trying to throw words out here for no reason. I want to review what all these things are because then we're going to talk about how these players interact. So just hang with me for a little bit. Okay, so amino acids. If you'll remember in chapter 3, we learned that there are 20 different types of amino acids. And those are just the building blocks or monomers that we put together to make proteins. And so one of our examples here is glutamine, for instance. That is one of the 20 amino acids. And remember, when we link those amino acids together, that is the primary structure of a protein. And so it's going to be important, the chemical nature of these various amino acids, because how they interact is then going to determine the secondary and quaternary and tertiary structures of these proteins. So right now, I just want you to remember that there are 20 different types of amino acids. They can be used in all kinds of different orders to make an array of various proteins. So this is an overview of how we make proteins within the cell. And there are 2 different parts to making proteins. One is transcription and one is translation. So we begin with our DNA, which we know codes for all of our proteins in our cell. And what transcription is basically unwinding that DNA. And we have our template strand and our complementary strand. And remember, this is all taking place in the nucleus. And what happens is we then create a strand of messenger RNA that is complementary to our DNA template. So we have our C that lines up with our G, our adenine that lines up with our T, and remember, we don't have T's, so we have a uracil that lines up with our adenine, again our guanine with our cytosine, our cytosine with our guanine, and again with our guanine. And here we have 3 uracils that complement the adenine, because we are making a complementary strand that temporarily binds with the DNA template. And remember, those are hydrogen bonds, easy to break. Okay. Each of 3 nucleic acids make up what we call a codon. And that codon, CAU, GCC, UUU, each one is specific to an amino acid. Now, having said that, there are multiple codons that code for the same protein. So there is redundancy within our system so that if we have any damage, we can still produce the proteins that we need to. So we've done transcription. Transcription is taking DNA and creating a messenger RNA, and all that happens in the nucleus. Then the messenger RNA is sent to the ribosomes. And this next portion is what we call translation. So it's basically saying, okay, here's our code, we're going to decode it and make a protein from it. So like I said, each one of these codons then codes for a particular amino acid, and they're made in order along the mRNA, and they code for a particular amino acid. So CAU, for instance, codes for histidine. This is one of our amino acids. And so we denote amino acids usually by a 3-letter grouping. So HIS is for histidine, ALA is alanine, PHE is phenylalanine. Okay, so this again is translation, and it takes place in the ribosome. Okay, so here are the ones that we just, the amino acids that we just produced. Okay, so here is our phenylalanine, our alanine, and our histidine. Okay, so they have specific chemical structures, and that's going to become important. So this should be a little bit of a review. Here's our primary structure of our protein, and that's what we have just produced. Each of these little beads represents one amino acid. So we can pretend in here that we have our histidine, alanine, and phenylalanine. And so that's our primary structure. Now, based on those chemical formulas of those amino acids, then we get our secondary structure. And remember, that can be an alpha helix or it can be a beta pleated sheet. From there, then we go into our tertiary structure, and that has to do with all kinds of different chemical groups within our amino acids and how they interact with each other. That can then have our tertiary structure, and then multiple ones joined together for a quaternary structure, if that's going to be the destiny of that particular protein. So when we look at the codons always have specific amino acids, like I said. And there are certain codons that are start codons and stop codons. A start codon means, okay, you need to start making the protein here. This is where the code is written for the protein that we're looking for. And this table right here gives you a whole bunch of codons and what they code for. No, you don't have to memorize all of those, but I do want you to know the start codons and stop codons in particular. So if we look on this chart and we are looking for start codons, we find that there is only one. So that is actually, let me see if I can get my pen here. It's right here. AUG. And that's the mRNA. So our nucleic acid, A, U, and G, adenine, uracil, and guanine. That is always the start codon, and there's only one of them. And that start codon always codes for the amino acid methionine. So every protein starts with a methionine amino acid. And then there can be a series of codons, depending on how long the protein is going to be, how many amino acids are in it. And then there's always a stop codon. and it can be one of any of these three, UAA, UAG, or UGA. Those are the stop codons. That means it tells the enzymes that are involved in producing these amino acids to stop reading right there. And a lot of times they knock it off. They knock that particular protein off. But those are our stop codons, UAA, UGA, and UAG. Okay, now you have a lot of the terminology. So let's go back to, we said gene expression has two stages, transcription and translation. Transcription is making messenger RNA from the DNA, and translation is making proteins from that mRNA. Okay, so here is just another overview, a little more detailed. We have our double helix. It gets unwound, and we have the template DNA right here with its bases sticking out. And then this is transcription in the nucleus. We then have complementary mRNA. And note that each of the three pairs is a codon. And each codon, each three base pairs, then is translated at the ribosome into a particular amino acid. In this case, we have arginine, tyrosine, and tryptophan. And that, when they're bound together, makes a polypeptide, or a protein. Okay, so gene expression. Making a protein from a gene, we said, has two parts, transcription and translation. The transcription part is taking the DNA gene and making messenger RNA. So let's take a closer look at that. That right there is going to require an enzyme called RNA polymerase. Now that might sound familiar because when we were replicating DNA, we used a protein called DNA polymerase. So similar functions. The RNA polymerase is going to untwist and unwind the DNA. It's going to recruit the free nucleotides to the DNA and basically make an mRNA strand based on the DNA template. Okay, so let's take a closer look at the DNA gene itself. Okay, when we unwind our DNA, and this is our lovely DNA here in white. I know it doesn't look a thing like DNA, but use your imagination a little bit. We have, first of all, what's called a promoter sequence. The promoter has a sequence of nucleic acids that's found right before the part of the gene that needs to be transcribed. And so the promoter is the area that tells the RNA polymerase where to start. And that's actually really important because if you think about how our genes are coded, it's a sequence of 3 nucleic acids at a time. Now, if our RNA promoter is off and it starts on, say, the second nucleic acid, well, that can mess up the entire series. So this promoter portion is actually really important. So the promoter is where the RNA polymerase begins. Then there's the transcribed region, and that's the DNA sequence that the RNA polymerase reads and uses to make the mRNA strand. And then there is what we call a terminator sequence, and that is exactly what it sounds like. It's a DNA sequence that comes after the transcribed region, and that tells the RNA polymerase to stop reading the genes right there. So that's sort of an overview of what our DNA looks like for the part that we are going to transcribe into a protein. So here is our overview again. We have our DNA, this lovely stringy thing that for some reason in this image looks like an earthworm. I don't know why, but okay, there's our DNA sequence, and we have our promoter right here that tells the RNA polymerase that it can start transcribing. We have our transcription region and our terminator, and that's transcribed into a messenger RNA strand. Now this image also mentions what we call a regulatory sequence. And basically that is just a place that regulatory proteins bind, and the role of those regulatory proteins is to basically determine how fast this region is transcribed. And it doesn't always have to be right after the promoter. It can be throughout the transcription region. But if you remember the importance of homeostasis within a cell, these regulatory proteins play a role in that, because if you need, for instance, to produce insulin and you need it at a high rate, those regulatory proteins can come in and say, okay, let's make this faster. We got to get more of these produced. Consequently, if we have plenty of insulin, oftentimes the insulin will bind a regulatory protein that together binds the regulatory sequence and says, okay, stop making this. We don't need so much anymore. Okay? So that's what that regulatory sequence is for. So here's our picture of our RNA polymerase. That's that big enzyme that's going to transcribe the DNA to messenger RNA. And so in this particular image, our blue DNA is opened up, and we have the template strand on the bottom here. And in this case, the complementary strand really doesn't do anything. We don't need that right now. It's just sitting there as a placeholder. And so RNA polymerase then begins to build a messenger RNA by recruiting these RNA nucleotides, and we'll talk about a little bit more about that, to come in. And for each codon, so we're putting in complementary messenger RNA. And so remember, each of these three is going to be a codon that's then going to recruit an amino acid. But right now the RNA polymerase is making new messenger RNA, and it does have a direction of transcription. So here we see that RNA is built in the 5' to 3' direction, just like when we did DNA replication. Okay, so that's always going to be the case, but for this time, remember, we're using only the complementary strand. So if we aren't using that other strand, then we don't need to worry about things like Okazaki fragments like we did with DNA replication, because we're only using one strand of the DNA, and we can make it in make our messenger RNA in a 5' to 3' direction, nice and smooth. Okay, so big picture, when we are creating proteins, remember we said we have transcription process and translation. Okay, now we are going under transcription, and there are 3 steps to the transcription part. And this is pretty easy. It's pretty intuitive. We have initiation, elongation, and termination. And so what that means is the initiation step of transcription is when the RNA polymerase finds the promoter and begins transcription. The elongation step to transcription is basically when the mRNA is being made. Okay, so it's being transcribed. And then the third is the termination. The RNA polymerase reads the terminator sequence and falls off the DNA template. Okay, so that's the 3 parts to basically making messenger RNA, which is what transcription is. So just to show you how important that one enzyme is, we have poisonous mushrooms called the death cap mushrooms. And so this is why it's important when you're going through the forest. You know, some people go, oh, we can eat mushrooms, and ha ha ha, we have hallucinogens, and okay, that's lovely, but what about you knowing your mushrooms? Because death cap mushrooms inhibit the activity of RNA polymerase within liver cells. And they do that by binding the RNA polymerase in a way that means that the RNA polymerase cannot bind the DNA. So if we're not binding DNA, we don't have transcription occurring, and therefore we are not making the appropriate proteins within the liver cell. And so people who eat the death cap mushrooms tend to die within 10 days because this one protein is inhibited. So that should tell you the importance of RNA polymerase. Okay, so what is the end result of transcription? The end result of transcription is an mRNA that's like a rough draft. It's called the primary transcript. And it goes through processing, RNA processing, to fix that rough draft. In other words, it's going to find out if there are any mistakes and fix those. And that only happens in eukaryotes. And remember, we had this very similar thing with our DNA polymerase, and that processing of DNA only happens in eukaryotes. So aren't we glad that we are eukaryotes? Okay, so there are 3 steps that go into this RNA processing. So the very first thing that's going to happen to the messenger RNA is an addition of what we call a 5' cap. And so that is an addition of a guanine triphosphate molecule added to the 5' end. So what does that do? Okay, that cap allows the ribosome to recognize the messenger RNA. In other words, the ribosome that's going to produce the proteins has to be able to recognize our messenger RNA that comes out of the nucleus. So that 5' cap is what allows the ribosome to recognize it. The second thing that mRNA processing involves is the addition of what we call a poly-A tail. And so that is put onto the 3' end of our messenger RNA, and it's anywhere between 150 to 200 additional adenines. Why would we do that, right? Well, the function is that it helps the messenger RNA get out of the nucleus and keeps it from being degraded. Okay, so it's sort of like a signal part of the messenger RNA that takes it out of the nucleus and protects it from being degraded. And then there's the third part of RNA splicing, and this I find completely fascinating. So when we look at the messenger RNA, it has what we call exons and introns, different portions throughout the messenger RNA. And you can see those pretty well in our diagram over here. So we have exons and introns. And the exons are the parts of the messenger RNA that we keep. Those are the ones that are what we call expressed or translated into amino acids. And then the introns are the parts that are going to be removed. It's almost like they're fillers within the messenger RNA, and those are excised out. We take those out of the messenger RNA. Now, just something fun to think about is, why do we have introns at all? I think it's a great question. I don't know the answer. I don't think anybody knows the answer. Introns have always been thought of as portions of the messenger RNA that are just junk and thrown away. That's lovely to think of, but the highest organisms all have introns, and those lower organisms, the way we think of it, lower organisms, like our bacteria, don't have introns. So I feel like they must serve some purpose that we just don't know about yet, which is always the exciting part of biology, is that there's always something new to learn, because even at the cellular level, there's so much we don't know yet, which keeps it exciting. Okay, so we are going to take away those introns and remove them. What removes those introns? Lovely name. I always love how these enzymes are named. They're called the spliceosomes, and it's actually more than one protein put into a complex of proteins that recognize those intron sequences and cut them out, and then they splice together the exons. So it has to be a multi-protein complex, right? Because that's a lot of functions. So we call that the spliceosome complex. Okay, so here we go again with our worm-looking DNA. We have our promoter right here. We know that's where our RNA polymerase starts. And then we have our transcribed region, which is full of exons and introns, and then we have our terminator region. And we know somewhere in here we also have our regulatory sequence that our regulatory proteins bind as well. So in transcription then, remember this happens within the nucleus, we create a messenger RNA or a rough draft of the messenger RNA in the 5' to 3' direction, and it still has the introns in it. And then we go through the processing. Step 1, we cap the protein. Step 2, our splice enzymes come in. And step 3, we put a poly-A tail on it. So that at the end, we have a 5' to 3' messenger RNA that has a 5' cap, a 3' tail, and only exons within the messenger RNA. Okay, so here's our summary again of our overview on making proteins. We've just gone through the transcription part that all takes place in the nucleus, and at the end of it we come out with messenger RNA that has had the introns removed, a 5' cap put on, a poly-A tail put on, in order for it to be able to go from the nucleus out to the cytoplasm. And interestingly enough, I didn't mention before, all of those RNA processing steps, they help to basically allow for the mRNA half-life to be much longer than those in prokaryotic cells. So it can last for several hours in the cytoplasm. Okay, but the next part that we're moving on to is translation. Okay, so translation happens in the ribosome. So we are taking mRNA codons and producing amino acids in order to line them up to make a protein.